Hello everyone and welcome to today's presentation. I'm excited to be here with you. Today we'll be exploring Paclitaxel, nature's cancer fighter. From its origins in the ancient Pacific yew to its revolutionary role in modern cancer treatment, we'll see how this remarkable natural compound has transformed oncology. Over the course of this talk, I'll guide you through its fascinating history, how it works, and its impact on medicine today. Let's get started. Let's start by looking at the chemical identity and structure of paclitaxel. It's also known by its common names, with the brand name Taxol, which many of you might be familiar with, and its international non-proprietary name, or IN, paclitaxel. Its chemical identifiers include the NSC number 125973, which is used in research contexts. In terms of its molecular profile, paclitaxel has a complex formula, C47H51NO14, and it weighs approximately 853.91 grams per mole. It belongs to the class of compounds known as diterpenoids. These are large, complex molecules derived from 4 isoprene units, which contribute to their biological activity. Now, looking at its natural classification, paclitaxel is a taxane diterpenoid. It's actually a pseudoalkaloid, meaning it shares some features of alkaloids but isn't classified strictly as one. It's naturally sourced from yew tree species, which are well known for producing a variety of bioactive compounds. Connecting this to our broader discussion, understanding the chemical identity and structure of paclitaxel helps us appreciate how its natural origins and complex molecular makeup enable its powerful role in cancer treatment. It's a prime example of how natural products can be transformed into effective pharmaceuticals. Let's talk about the biological sources of paclitaxel. The primary source is Taxus brevifolia, commonly known as the Pacific yew. The bark of this tree contains the highest concentration of paclitaxel, making it the main source historically used for extraction. However, there are alternative sources too. For example, Taxus baccata, or the European yew, provides needles that contain precursors necessary for semisynthesis. This means we can use parts of this tree to produce paclitaxel more sustainably and efficiently. In addition to harvesting from trees, biotechnology offers promising solutions. Techniques like cell suspension cultures and endophytic fungi can produce paclitaxel in a controlled environment. These methods are more sustainable because they reduce the need to harvest large amounts of bark from wild trees, helping us protect natural resources while meeting demand. Connecting this to our overall theme, understanding these sources is crucial for developing sustainable and scalable ways to produce this important drug. Let's take a look at the global distribution of these species. First, in the Pacific Northwest, you'll find T. brevifolia, which grows across Washington, Oregon, California, and even into British Columbia. This region provides the ideal climate for this species to thrive. Moving over to Europe and Asia, T. baccata is quite widespread, covering Europe, North Africa, and West Asia. Additionally, there are Asian species found in China, Korea, and Japan, highlighting the diversity within this group. Now, when we consider cultivation centers, India stands out with regions like Himachal Pradesh and Sikkim cultivating these species successfully. China also plays a significant role, especially in Yunnan and Sichuan. Moreover, European plantations are vital for the propagation and study of these trees. Overall, this distribution underscores how adaptable and widespread these species are, reflecting their importance both ecologically and economically across different parts of the world. Now, let's take a closer look at the chemical constituents involved in this process. First up is paclitaxel, which is the primary active compound known for its potent anti-cancer properties. It's the main ingredient that makes this extract so valuable in cancer treatment. Next, we have 10-deacetylbicatine III. This compound serves as a key precursor, especially in semisynthetic production methods. It's an essential building block that allows us to produce paclitaxel more efficiently. Then there's baccatin III, which is an important intermediate in the taxane family of compounds. This intermediate plays a critical role in the synthesis pathway, bridging the raw material and the final active drug. Finally, we need to be aware of the taxane alkaloids. These are toxic constituents mainly found in the leaves of the plant. While they are part of the plant's natural chemistry, they pose a risk if not properly managed during extraction and processing. Understanding these constituents helps us appreciate the complexity of the plant's chemistry and why careful extraction and processing are so vital. It's about maximizing the beneficial compounds like paclitaxel while minimizing the risk posed by toxic constituents. Let's now look at the various identification methods used to analyze our compound. First, TLC analysis is a common technique. Here, silica gel is used as the stationary phase, with a solvent system of chloroform and methanol in a 9:1 ratio. When we develop the plate, purple-blue spots appear after spraying with vanillin sulfuric acid, indicating the presence of specific components. Next, we have HPLC detection. We use a reverse phase C18 column for separation, and detection is performed with UV at 227 nanometers. The retention time we observe helps us confirm the identity of the compound by matching it with known standards. Moving on to LCMS confirmation, this technique offers high specificity. We look for the molecular ion peak at M plus H plus equals 854.3 M. The characteristic fragmentation pattern associated with this peak provides further confirmation of the compound's structure. Finally, classical tests like the Lieberman-Burchard test are also employed. A positive result here suggests the presence of a diterpenoid structure, which aligns with what we expect for this compound. Altogether, these methods, TLC, HPLC, LCMS, and classical tests, offer a comprehensive approach to accurately identify and confirm our compound, tying back into our overall theme of thorough analytical characterization. Now, let's take a closer look at how paclitaxel works at the molecular level. It specifically binds to beta-tubulin subunits, which are essential building blocks of microtubules. By attaching here, paclitaxel stabilizes these microtubules, preventing them from breaking down or depolymerizing. This stabilization is crucial because it disrupts the dynamic nature of microtubules that cells need for division. As a result, cells become stuck in the G2M phase of the cell cycle, essentially putting a halt to their division process. This arrest then triggers apoptosis, or programmed cell death, which is how paclitaxel exerts its anti-cancer effects. So, in summary, by binding to beta, tubulin, and stabilizing microtubules, paclitaxel effectively blocks cell division and promotes cancer cell death. Let's take a closer look at some of the key clinical applications of this treatment. First, in ovarian cancer, it's commonly used as a first-line treatment and often combined with carboplatin. This combination helps to improve the overall efficacy, giving patients a better chance at controlling the disease early on. Moving on to breast cancer, this therapy is versatile. It's used both in early-stage cases and in metastatic settings, offering benefits across different stages of the disease. In lung cancer, specifically non-small cell lung cancer, this has become a standard part of combination regimens. It's really become a cornerstone in the treatment for this type of lung cancer. And finally, for Kaposi's sarcoma, particularly in AIDS-related cases, this therapy has shown to be quite effective. It's an important option for patients facing this type of cancer linked to immunosuppression. Overall, these applications highlight the broad and impactful role this treatment plays across various cancers, emphasizing its significance in personalized cancer care. Let's take a look at the market preparations for these important drugs. First, we have the brand name, formulation, and manufacturer details. For example, Taxol Registered is a well-known brand of paclitaxel injection, produced by Bristol-Myers Squibb. Then there's Abraxane Registered, which is albumin-bound paclitaxel developed by Celgene in the USA. This formulation is particularly interesting because it improves delivery and reduces some side effects compared to traditional versions. We also see Onxal Registered, which is a generic paclitaxel made by multiple producers, making it more accessible and often more affordable. Another notable product is NanoZell Registered, created by Dabur Pharma in India, utilizing a nanoparticle formulation. This approach enhances drug delivery and potentially improves efficacy. Then there's Taxol registered, produced by Fresenius Kabi in India, which is available as paclitaxel injections. These come in various forms: standard IV injections, nanoparticle suspensions, and liposomal formulations, each designed to improve delivery and effectiveness. Overall, what this slide highlights is the diversity of formulations and manufacturers, reflecting how the market has expanded to include different delivery systems and improved drug designs. This variety is crucial for tailoring treatments to patient needs and optimizing therapeutic outcomes. Let's talk about the market potential and sustainability aspects of paclitaxel. Currently, the global market value for paclitaxel exceeds $5 billion, which highlights its significant role in cancer treatment worldwide. The market is growing at about 25% annually, mainly driven by rising cancer rates and the increasing use of combination therapies to improve patient outcomes. However, there's an important sustainability challenge we need to address. Traditionally, paclitaxel has been harvested from the bark of Pacific yew trees, and this process can be harmful to the environment because it kills the trees. This creates environmental concerns that we can't ignore. Fortunately, there are promising future solutions emerging. Semi-synthetic production methods and the use of plant cell cultures are being developed as more sustainable alternatives. These approaches can help us meet the growing demand while also protecting our ecosystems. Overall, understanding both the market potential and the sustainability challenges is crucial as we look toward innovative ways to improve access and environmental impact in cancer therapies.